From the moment we enter the world, our bodies are surrounded by a swirling sea of microscopic life. Bacteria, viruses, and other microbes drift through the air, linger on surfaces, and sometimes even live quietly on our skin. Most of these tiny organisms are harmless, some are even beneficial. But others, pathogens, have the potential to make us very sick. Fortunately, our bodies are not defenseless. Every second, a sophisticated network called the immune system stands guard, working tirelessly to recognize and eliminate threats before they can harm us. The immune system is a marvel of biological engineering. Its first layer of defense is physical. The skin, mucous membranes, and cilia lining our airways act as barriers, blocking many germs from ever entering the body. If a pathogen manages to get through, the immune system springs into action with two main strategies: innate and adaptive immunity. The innate immune system, which includes cells like macrophages, responds rapidly and indiscriminately, attacking any foreign invader it encounters. This non-specific defense buys time for the adaptive immune system to mobilize, a more specialized, targeted response that can remember specific pathogens and eliminate them more efficiently if they return. At the heart of the adaptive immune system are two types of white blood cells: T lymphocytes and B lymphocytes. When a pathogen invades, its unique surface molecules, called antigens, are recognized as foreign. Macrophages and other antigen-presenting cells digest the invader and display fragments of these antigens on their surfaces. This display signals T cells, which coordinate the immune response, and B cells, which begin producing antibodies, proteins that bind specifically to the pathogen's antigens and help neutralize or destroy it. Some of these B and T cells transform into memory cells, standing ready for years or even decades to respond with lightning speed if the same pathogen tries to return. This ability to remember is one of the immune system's most powerful traits. It is also the foundation upon which vaccines are built. Vaccines are ingenious tools that train the immune system, safely imitating infection so our bodies can practice defense before facing the real threat. Instead of waiting for a dangerous germ to invade, vaccines introduce harmless pieces of a pathogen, or, in some cases, a set of instructions that allows our cells to briefly make those pieces, so the immune system can learn to recognize and fight the invader in advance. There are several different types of vaccines, each offering a unique way to present antigens to the immune system. Some, known as live-attenuated vaccines, contain a weakened form of the actual pathogen. These weakened germs usually do not cause disease in healthy people, but they are similar enough to the real thing to provoke a strong, lasting immune response. Live, attenuated vaccines often provide long-term immunity with just 1 or 2 doses, but they are not suitable for everyone. For example, people with weakened immune systems should avoid them. In contrast, inactivated vaccines use germs that have been killed or inactivated, so they cannot replicate or cause disease. While these vaccines are very safe, especially for people with underlying health conditions, the immune response they generate is sometimes less robust, which means booster shots may be needed over time to maintain protection. Then there are subunit, recombinant, polysaccharide, and conjugate vaccines. These use only specific parts of the germ, often fragments of protein or sugar molecules that the immune system can easily recognize. By focusing on these key antigens, these vaccines reduce the risk of side effects, but they often require multiple doses to achieve strong, lasting immunity. Toxoid vaccines take a slightly different approach. Some bacteria cause disease not by invading our tissues, but by releasing toxins. Toxoid vaccines contain inactivated toxins, called toxoids, that cannot cause harm but still train the immune system to recognize and neutralize the real toxin if it ever appears. Classic examples include the vaccines for diphtheria and tetanus. In recent years, advances in science have paved the way for entirely new vaccine technologies. Messenger RNA, or messenger ribonucleic acid, vaccines represent a major innovation. Instead of containing parts of the pathogen itself, these vaccines deliver a blueprint, a messenger RNA sequence, that instructs our cells to briefly make a harmless piece of the virus, usually a protein found on its surface. The immune system then mounts a response to this protein, creating antibodies and memory cells without exposure to the actual pathogen. Messenger RNA vaccines have been used effectively against COVID-19 and offer the promise of rapid development for future threats. Viral vector vaccines use a different kind of delivery system. They employ a harmless virus, modified so it cannot cause illness, to carry genetic instructions into our cells. These instructions prompt the cells to produce antigens from the target pathogen, again stimulating an immune response. This technology has been used in some Ebola and COVID-19 vaccines. As scientists continue to explore new approaches, Other forms, like virus-like particle vaccines, synthetic peptide vaccines, and DNA vaccines, are being developed and tested for a variety of infectious diseases. Each type of vaccine has benefits and limitations, and researchers select the best approach based on the disease, the target population, and the practical challenges of manufacture and distribution. No matter the type, all vaccines work on the same basic principle. They provide the immune system with a safe preview of a pathogen, allowing it to learn without the risk of severe illness. After vaccination, the immune system responds by producing antibodies and memory cells, just as it would after a natural infection. The key difference is that vaccines allow this training to happen without the dangers associated with the actual disease. The immune response to vaccines is a complex dance involving many types of cells and molecular signals. When antigens from a vaccine are introduced into the body, they are picked up by antigen-presenting cells, such as macrophages. These cells process the antigens and display them for T cells to see. Helper T cells then activate B cells, which begin to churn out antibodies specific to the antigen. Meanwhile, cytotoxic T cells can seek out and destroy cells infected by the pathogen if needed. The immune system also releases cytokines, chemical messengers that help coordinate the response. Most importantly, some of the activated B and T cells become long-lived memory cells. These memory cells patrol the body for years, ready to respond swiftly if they ever encounter the same antigen again. This rapid response is why people who have been vaccinated usually do not get sick or experience only mild symptoms if they are exposed to the real pathogen later on. It is important to note that the strength and duration of immunity can vary depending on the vaccine and the individual. Live-attenuated vaccines tend to produce longer-lasting immunity, while inactivated or subunit vaccines may require boosters. Some people may respond more strongly to vaccination than others due to differences in genetics, age, or health status. Vaccines do not just protect individuals, they can also help shield entire communities by reducing the spread of disease. When enough people are immune, either through vaccination or prior infection, it becomes much harder for a pathogen to find new hosts. This phenomenon, known as herd immunity, helps protect those who cannot be vaccinated, such as infants or people with certain health conditions. Most vaccines provide active immunity, in which the body's own immune system is prompted to respond and remember. Others, such as the antibodies passed from mother to child before birth, offer passive immunity, temporary protection without the creation of memory cells. While passive immunity fades over time, active immunity from vaccines can last for many years. The science of vaccines is constantly evolving. Researchers continue to seek better ways to stimulate the immune system, target tricky pathogens, and tailor vaccines to the needs of different populations. New technologies such as messenger RNA and viral vector platforms have expanded our toolkit and hold promise for the future. At its core, vaccination is a simple but profound idea. By teaching our immune system to recognize and remember the enemy in advance, we gain the power to prevent disease, protect our communities, and save millions of lives. In this ongoing partnership between human ingenuity and biology, vaccines stand as one of the greatest achievements in the fight against infectious diseases. Imagine a time when the threat of infectious disease was a constant looming presence. When smallpox alone haunted nearly every corner of the globe, claiming lives and leaving survivors marked or even blinded. The story of vaccination is, in many ways, the story of humanity's long struggle to outwit such invisible enemies. Our journey begins not in the laboratories of brilliant scientists, but centuries earlier, in the hands of healers and communities who observed, experimented, and shared knowledge in hopes of staving off disaster. Long before the word vaccine entered our vocabulary, societies in China had developed a practice now known as variolation. As early as the 15th century, Chinese healers exposed healthy individuals to powdered material taken from the scabs or pustules of people recovering from smallpox. The hope was that a mild infection would result, conferring protection against the far deadlier full-blown disease. This early form of immunization spread along trade routes, eventually reaching the Ottoman Empire. In Istanbul, variolation was widely used by the early 18th century, and it was here that Lady Mary Wortley Montagu, the wife of the British ambassador, encountered the practice. Having witnessed the devastation of smallpox firsthand, she recognized its potential for prevention. She arranged for her own children to be variolated and, upon returning to Britain, advocated for its adoption in England. The idea was radical and the technique controversial. Yet the urgency of epidemics, such as the one that swept through Boston in 1721, forced communities and physicians to weigh the risks. In colonial America, the introduction of variolation was met with both hope and suspicion. A particularly striking episode occurred during the American Revolutionary War, when George Washington ordered the inoculation of his troops. The risk of losing soldiers to smallpox was too great to ignore, and this decision is now widely considered a turning point in both military and medical history. The leap from variolation to vaccination, a safer and far more effective intervention, came at the close of the 18th century. Edward Jenner, an English physician, observed that milkmaids who had contracted cowpox, a disease similar to but much milder than smallpox, seemed immune to smallpox itself. In 1796, Jenner tested his hypothesis by taking material from a cowpox sore on a milkmaid's hand and introducing it into the arm of a young boy named James Phipps. The boy developed a mild illness but did not contract smallpox, even after subsequent exposure. Jenner's experiment is often hailed as the birth of modern vaccination, and the very word vaccine is drawn from vacca, the Latin word for cow. Jenner's method quickly gained traction. By the early 1800s, vaccination against smallpox had spread across Europe and to the United States. The first vaccinations in America took place in 1800, and public health authorities gradually began to recognize the power of this new tool. In 1855, Massachusetts became the first United States state to mandate smallpox vaccination for schoolchildren, a move that signaled both growing confidence in vaccines and the enduring challenge of balancing personal liberty with public health. The story of vaccination, however, does not belong to Jenner alone. As the 19th century unfolded, Louis Pasteur, a French chemist and microbiologist, took up the challenge of developing vaccines against other deadly diseases. Pasteur's approach was rooted in the emerging science of microbiology. He understood that diseases were caused by specific microbes and believed it was possible to weaken these agents in the laboratory. His work led to the creation of the first laboratory-produced vaccines, starting with chicken cholera and anthrax. Pasteur's crowning achievement came in 1885 when he tested a rabies vaccine on a young boy named Joseph Meister, who had been bitten by a rabid dog. The boy survived, and the world witnessed the transformative potential of vaccination in real time. The late 19th and early 20th centuries were an era of discovery and innovation in vaccinology. The principles established by Jenner and Pasteur inspired a generation of scientists. New vaccines followed for diseases such as diphtheria, tetanus, and pertussis, each one a technical and logistical challenge, and each a triumph over a long-feared illness. The 20th century, in particular, saw an explosion of progress, with the advent of vaccines for yellow fever, polio, measles, and mumps. These breakthroughs were often the result of international collaboration and fierce determination. Jonas Salk and Albert Sabin, for example, became household names for their work on polio vaccines, one using an inactivated virus and the other a live, attenuated strain. Their efforts, supported by widespread immunization campaigns, brought the world to the brink of eradicating polio, a goal that remains tantalizingly close today. Other pioneers pushed the boundaries of what vaccines could achieve. Maurice Hilleman, though less known to the general public, was responsible for developing over 40 vaccines, including those for measles, mumps, hepatitis A, hepatitis B, and meningitis. His work has saved countless lives and continues to shape immunization schedules around the world. Stanley Plotkin, another leading figure, made foundational contributions to vaccines for rubella and rabies, and his influence is felt in the ongoing refinement of vaccine science. With each new vaccine, societies confronted questions of access, acceptance, and oversight. In the United States, the expansion of vaccine mandates, such as the Massachusetts school entry requirement, was accompanied by efforts to track and measure vaccination rates. Public health surveillance became an essential tool for monitoring the success of immunization programs and identifying gaps in coverage. The true measure of vaccination's impact is most dramatically illustrated by the fate of smallpox. For centuries, this disease was a universal terror, killing or disfiguring millions. After nearly 2 centuries of vaccination, the World Health Organization declared smallpox eradicated in 1980. This was the first, and so far, only human disease to be eliminated from the planet through vaccination alone. The eradication of rinderpest, a devastating cattle disease, followed in 2011, and the near eradication of polio stands as a testament to sustained international effort. Yet the history of vaccines is not only a story of triumphs. New challenges have emerged as science has advanced. The development of vaccines for chronic infections, such as HIV and malaria, has proven far more difficult than for acute diseases. Scientists have responded with innovative strategies, including the use of adjuvants to boost immune responses, prime boost regimens that combine different vaccine types, and a growing focus on mucosal immunity, which targets pathogens at the body's entry points. The pace of innovation has only accelerated in recent decades. Advances in virology and molecular biology have enabled the creation of recombinant vaccines and, more recently, the rapid development of messenger RNA-based vaccines. The COVID-19 pandemic showcased the speed and flexibility of these new platforms, ushering in a new era for immunization and disease prevention. Throughout this history, the language of vaccination has evolved alongside the science. Early terms like inoculation and variolation have given way to a lexicon that reflects deeper understanding and broader applications. What remains constant, however, is the underlying principle. By harnessing the body's own defenses, vaccines offer protection not only to individuals but to entire communities. Today, the legacy of those early experiments and the vision of pioneering scientists are visible everywhere, from the vials in a clinic refrigerator to the global efforts to track, deliver, and improve vaccines for future generations. The journey from the dusty streets of ancient China to the gleaming laboratories of modern medicine is a testament to human ingenuity, persistence, and the enduring hope that diseases which once defined the limits of life can be consigned to history. Each new vaccine adds another chapter to this unfolding story, reminding us of what is possible when knowledge, courage, and compassion converge in the service of health. Vaccines are not just a medical marvel. They are a dynamic force in the ongoing battle against disease, shaping the very fabric of public health worldwide. Their impact is measured in millions of lives saved, communities protected, and outbreaks averted. Yet, their story is ever evolving. It unfolds daily in clinics, remote villages, and bustling cities, where the promise of immunization faces both victories and persistent challenges. Consider the scale of their influence. Each year, routine childhood vaccination is credited with preventing about 4 million deaths globally. The World Health Organization estimates that immunization programs now avert between 3.5 and 5 million deaths annually, shielding people from more than 30 life-threatening diseases. These numbers are not just statistics. They represent children who grow up instead of succumbing to pneumonia or measles, parents spared the anguish of losing a child, and communities able to thrive without the constant threat of epidemic disease. Yet, the toll of vaccine-preventable illnesses remains sobering. In 2018 alone, an estimated 700,000 children under the age of 5 died from diseases that could have been prevented with existing vaccines. Most of these young lives were lost in low- and middle-income countries, where Streptococcus pneumoniae, rotavirus, pertussis, measles, Haemophilus influenzae type B, and influenza continue to exact a tragic price. The power of vaccines becomes even clearer when we look at their real-world effect on disease incidence. Decades ago, measles and pertussis were common childhood illnesses, sometimes leading to death or lifelong disability. Today, where vaccines are widely used, these diseases have become rare. Yet, their absence can lead to complacency. As immunization rates decline even slightly, outbreaks quickly resurface, reminding us how fragile disease control can be. In recent years, resurgences of measles and pertussis have been reported not only in regions with limited resources, but also in high-income countries, including North America. These outbreaks are often traced to pockets of low vaccination coverage, where the protective shield of community immunity has weakened. This brings us to a concept at the heart of vaccination's public health power: herd immunity, sometimes called community immunity. When a sufficient proportion of people in a population are immune to a disease, usually through vaccination, the spread of that disease is effectively blocked. The microbe struggles to find new, susceptible hosts, and even those who are unvaccinated, such as infants, people with certain medical conditions, or the elderly, are indirectly protected. The threshold required for herd immunity varies by disease. For highly contagious infections like measles, upwards of 95% of the population must be immune to halt transmission. For others, the threshold is lower. Achieving and maintaining this level of immunity is a complex task, influenced by vaccine effectiveness, community structure, and patterns of contact between people. The story of herd immunity is not just one of percentages and statistics, but of practical, lived reality. In communities where vaccination rates remain high, outbreaks are rare and quickly contained. But when those rates slip, old adversaries return. The COVID-19 pandemic underscored both the promise and the challenge of herd immunity. While the concept offered hope for controlling the virus, the World Health Organization made it clear that herd immunity should be achieved through vaccination, not by allowing unmitigated spread of disease. Relying on natural infection can have catastrophic consequences, especially for the most vulnerable. The pandemic also revealed the importance and the difficulty of maintaining high levels of immunity when access to vaccines is unequal or disrupted. Vaccines do more than protect individuals. They transform societies. In the poorest countries, expanding access to vaccines creates a ripple effect that extends far beyond health. Each dollar spent on immunization in low and middle-income countries is estimated to save $52 by preventing medical expenses, lost productivity, and the broader economic drain of illness. Since the dawn of modern immunization programs, vaccines have saved an estimated 154 million lives, and routine vaccination alone has prevented more than 20 million deaths. These achievements are the result of coordinated global efforts, persistent outreach, and the tireless work of health workers who bring vaccines to the remotest corners of the world. Yet, the goal of universal immunization remains elusive. In 2023, more than 14.5 million children received no vaccines at all, while another 22 million missed key doses. Globally, 1 in 5 children lacks access to basic vaccines. The reasons are as varied as they are stubborn. Some children live in areas plagued by conflict or fragile health systems. Others fall through the cracks of underfunded programs or face logistical barriers like the lack of a reliable cold chain to keep vaccines effective. In middle-income countries, coverage gaps and inequities persist, highlighting that access is not just about wealth but also about political commitment, infrastructure, and sustained public health investment. The coronavirus disease 2019 pandemic further exposed and deepened these challenges. Routine immunization coverage plateaued and, in many places, fell below pre-pandemic levels, particularly for critical vaccines like diphtheria, tetanus, pertussis, and measles. Fragile settings, where health systems are already stressed, were hit hardest. Even as the world races to catch up, the need for ongoing system strengthening is clear. Barriers to access are not limited to remote villages. They exist in underserved urban neighborhoods and marginalized communities everywhere. Efforts to address these obstacles focus on equity, ensuring that no child, regardless of geography or circumstance, is left behind. With such high stakes, it is no surprise that questions and concerns about vaccine safety are common. Decades of rigorous research have built a robust foundation of evidence. Modern vaccines are among the safest interventions in medicine. The vast majority of side effects are mild and temporary: soreness at the injection site, a low-grade fever, or fatigue. Severe adverse reactions are exceedingly rare, and when they do occur, they are carefully investigated. Multiple large studies have demonstrated that vaccines do not cause autism, a myth that has been repeatedly and thoroughly debunked. The notion that vaccines overload the immune system is not supported by scientific evidence. The immune system encounters far more antigens in daily life than in the entire childhood vaccine schedule. Still, misinformation and myths can erode public confidence. Some believe that diseases targeted by vaccines have disappeared, so immunization is no longer needed. In reality, these diseases persist and can return swiftly if vaccination rates drop. Others worry that vaccines are not effective, or that natural infection is somehow preferable. The scientific consensus is clear. Vaccines confer strong, lasting immunity without the risks of severe illness or death. Public health agencies and medical professionals continually monitor vaccine safety, update recommendations, and communicate transparently about benefits and risks. Yet, vaccine hesitancy remains a significant barrier. It can stem from distrust, misinformation, or past negative experiences with health systems. Addressing hesitancy is not simply a matter of providing facts, but of building trust, listening to concerns, and engaging communities in respectful dialogue. The stakes are high not just for individuals, but for entire communities. The resurgence of preventable diseases in areas where vaccine coverage has declined is a stark reminder that the success of immunization programs depends on sustained collective effort. Looking ahead, the challenges of global immunization are daunting, but not insurmountable. Funding priorities, affordability, and long-term commitment are all essential. The ongoing effort to eradicate polio, reduce measles mortality, and expand access to new vaccines in middle-income countries continues. The fight against malaria, tuberculosis, and HIV remains complex, but the tools and knowledge gained from past successes inform future strategies. Vaccines are more than a shield, they are a promise. A promise that, through science, cooperation, and vigilance, we can protect not just ourselves, but each other. The work is ongoing, and the stakes measured in lives, health, and hope could not be higher. The world stands at a turning point in the story of vaccines, a moment shaped by scientific ingenuity, hard-won lessons, and the daunting realities of global health. As researchers look to the future, the field is alive with promise, particularly from emerging technologies like messenger RNA or mRNA vaccines. These innovative biotherapeutics have already proven their power in the fight against COVID-19, but their true potential extends far beyond a single pandemic. Scientists are now investigating how messenger RNA vaccines could be used to combat a wider range of diseases, including some of humanity's most persistent adversaries, such as HIV, malaria, and even certain types of cancer. At the core of messenger RNA vaccine technology lies a simple yet profound idea. Instead of introducing traditional antigens or weakened germs, these vaccines deliver genetic instructions, short strands of messenger RNA that prompt our own cells to produce specific proteins associated with a pathogen. This process trains the immune system to recognize and attack the real threat if it ever appears. Because this method is modular and does not require growing live viruses or bacteria, messenger RNA vaccines can be designed and produced with remarkable speed and flexibility. This advantage was on full display during the COVID-19 pandemic, when messenger RNA vaccines were developed, tested, and authorized in record time. What excites researchers now is the remarkable adaptability of this platform. Evidence is mounting that messenger RNA vaccines could be tailored to target a wide variety of infectious agents, as well as non-infectious diseases. For example, current research is exploring messenger RNA vaccines aimed at viruses like HIV, and at generating immune responses against certain cancers by instructing the body to attack malignant cells. The hope is that, with further refinement, messenger RNA vaccines might one day enable highly personalized treatments, customizing each vaccine or therapeutic to an individual's unique genetic makeup or the specific mutations present in their disease. Of course, this transformative approach comes with its own set of challenges. One major obstacle has been the inherent fragility of messenger RNA molecules, which are prone to rapid degradation in the body. Researchers have responded with inventive solutions, such as developing hybrid nanoparticles that shield the messenger RNA payload and improve its targeted delivery to immune cells. These advances are more than technical feats. They are essential to expanding the reach and effectiveness of messenger RNA vaccines, making them more reliable and easier to use in a variety of settings. Another exciting frontier in vaccine innovation is the development of universal vaccines, formulations designed to provide broad protection against entire families of viruses rather than a single strain. Early-stage candidates are being tested for diseases like influenza and coronaviruses, with the goal of streamlining responses to future outbreaks. For instance, some research focuses on vaccine approaches that stimulate the immune system to prepare for a wide spectrum of related viruses, potentially reducing the need for annual updates or new shots whenever a novel variant emerges. Yet the promise of these innovations is only part of the story. For any breakthrough to make a real difference, it must reach people everywhere, not just those in wealthy nations. The COVID-19 pandemic exposed deep inequalities in vaccine access. Even as new vaccines were developed at unprecedented speed, many countries faced shortages, high prices, and logistical barriers that left millions unprotected. The global community now faces urgent questions. How can production be ramped up quickly enough to meet worldwide demand? Who gets priority access when supplies are limited? And how do we ensure that the cost of innovation does not put vaccines out of reach for those who need them most? These concerns are not new, but the stakes have never been higher. Ensuring global access to vaccines requires more than scientific expertise. It demands coordinated efforts in manufacturing, distribution, and financing. Production capacity must be expanded, supply chains strengthened, and regulatory hurdles addressed, especially in low and middle-income regions. There is also growing recognition that clinical trials must be more inclusive, enrolling people from diverse backgrounds to ensure that vaccines are safe and effective for everyone. Without such measures, the gap between innovation and impact will only widen. Research into future pandemic preparedness has highlighted several key strategies to address these challenges. One lesson from COVID-19 is the value of investing in vaccine platforms, technologies that can be rapidly adapted to new threats. By building flexible manufacturing infrastructure and maintaining networks for data sharing and surveillance, countries can mount faster, more coordinated responses when the next outbreak occurs. Some experts advocate for global agreements that prioritize equitable vaccine allocation, ensuring that access is not determined solely by wealth or geopolitical influence. The role of regulatory agencies and international organizations is also coming into sharper focus. Streamlined approval processes, clear safety and efficacy guidelines, and transparent communication are all essential to keep pace with the speed of scientific discovery. At the same time, recent experience has underscored the importance of ongoing monitoring, tracking side effects, adjusting recommendations, and building public trust through openness and accountability. Looking further ahead, the research landscape is brimming with creativity. New vaccine platforms are being explored, from microarray patches that deliver vaccines painlessly through the skin to artificial intelligence-driven design tools that predict the most effective antigens. Efforts are underway to create vaccines that remain stable at room temperature, eliminating the need for expensive cold storage and expanding the reach of immunization campaigns in remote or resource-limited settings. There's particular interest in developing vaccines that work for people who are immunocompromised, those whose bodies may not respond robustly to traditional formulations. Meanwhile, the field of personalized vaccines is gaining momentum, especially in cancer research. Early clinical trials are testing messenger RNA-based vaccines designed to train the immune system to recognize and destroy each patient's unique set of tumor markers. While these approaches remain experimental, their progress hints at a future where vaccines are not just tools for prevention, but also for highly targeted treatment. Yet none of these advances will fulfill their promise unless the world commits to equitable innovation. Patterns of funding, research focus, and access have too often favored diseases prevalent in wealthier regions while neglecting threats that loom largest in low- and middle-income countries. Addressing these disparities will require intentional investment, policy reforms, and sustained collaboration across borders. The benefits of vaccination, reduced mortality, stronger economies, and healthier communities are global, and so too must be the effort to realize them. As the next generation of vaccines moves from laboratory benches to clinics and communities, their success will depend on more than molecular breakthroughs. It will rely on the world's willingness to learn from the past, invest in the future, and place the needs of the many above the interests of the few. The innovations of today offer the tools to confront old scourges and new threats alike. But the greatest challenge remains ensuring that these tools are available to all, so that the story of vaccination continues not just as a tale of scientific triumph, but as a testament to shared responsibility and hope.